Hi, good afternoon. Thanks so much for the invitation to share with you what we are learning from the use of these non-human primate models of development. And here, the disadvantage that we have is that when we are talking about the effects of early life stress or early adverse experiences on development as a model for psychiatric disorders like anxiety, depression, and also addiction, is that one of the most devastating experiences that we can have is really have lose our parents, have a disturbed relationship with them, suffer maltreatment, physical abuse or neglect. So the alterations of the mother-infant relationship are one of the most powerful early adverse experiences. And I'm going to focus on a model that we have been exploring, and it has turned out to be very interesting, which is a naturalistic model of infant maltreatment. And unfortunately, what I'm going to be telling you a lot is about how we have gone about validating this model. So I don't have a lot of answers about treatment. This is a very complex problem, and we know that it is because it's, in humans is experiencing early adverse experiences and early life stress like poverty, childhood maltreatment, trauma is one of the main risk factors for the development of psychiatric illness. And as I said, is anxiety, depression, and addiction. And it's very timely that we have this conversation due to the opioid epidemic crisis. But it's one of the main risk factors also for a set of different problems, including dropout in school, criminal behavior, ending up in jail. And also there is evidence that is a main risk factor for development of Alzheimer's. So this is in humans, and the issue with the humans is, with the human literature and the human studies, is that, as I will show you, that I'm going to be talking a lot in the first slide and the last slide, because I have some testimonials from clinical researchers where they're going to be telling you in their own words how important it is that we do this work, because they don't know where to start. So they are basically in the dark, because they have a child that they know, or they assume that they have gone through trauma, through maltreatment, but many of these cases are under the radar. They don't make it to social services, they don't make it to the hospital, people don't report it, and you end up with retrospective evidence or recollection of trauma, but you cannot measure it, you cannot go to the houses of the people, you cannot study them, and it's very difficult to do prospective and longitudinal studies. So we have been trying to put a lot of effort developing a translational model. And I'm going to be also focusing on the main target of this set of conversations that we are having today, which is why do we need a non-human primate model to study this? Because there are some studies that have been developed in rodents, but as I will tell you, there are some things that just don't translate very well. So I'm going to be focusing then in a model of early adverse experience that we study at Yerkes, the Primate Center. And we focus on how it affects the development of brain networks that regulate stress, emotion, and reward. And I'm going to be focusing on these pathways because they are important for explaining the underlying neurobiological alterations that lead to psychiatric disorders like anxiety, emotion, dysregulation, etc. Oh, the other thing that I forgot to say is that we are also having like a double whammy in this field, because we don't have just one brain that is developing. We have two brains. We have the brain of the baby, and we have the brain of the mother. And I don't know how many of you know or remember Hofer's theory that really the mother is the external regulator of the baby. So the baby needs the mother as an external hypothalamus, external prefrontal cortex. So there is this effect that the mother plays on the development of the brain, that basically the baby is just not alone. So we are really studying a unit that we call the mother-infant unit. So try to model that. As I said, and I turn this because it's very strange angle. We know there is a strong evidence that early life stress is a huge main risk factor for the development of psychiatric illness, in particular anxiety, depression, drug abuse, also cardiovascular disease, diabetes, obesity, and there are a set of different experiences that can lead to that, including poverty and infant maltreatment, which is the one that I'm going to be focusing on today. And that has different components, right? It can be physical abuse, sexual abuse, it can be neglect. And one of the things that has happened in this field is that we have been working under the assumption that these experiences are stressful and that they lead to the activation of stress systems, and therefore maybe stress hormones are mediating these effects. And there is a lot of biological explanations or linkage between elevations of stress hormones like cortisol and alterations in gene expression in the brain. But the truth is that the evidence is not there. This has been an assumption, but we don't have the hard data to demonstrate what are the actual biological mechanisms. And right now, in the last 10 years or so that we have been working on this, we do know that stress hormones are part of the story, but not the complete story. And I'm going to be telling you about other mechanisms that we have been publishing, including increased inflammation. And of course, we have all the evidence on epigenetic alterations that can be transmitted from one generation to another, which makes the story even more scary. As I said, one of the problems in this field is not only that you have two brains, you have this unit, mother-infant unit, is that you have a developing brain that we don't understand the adult brain completely, right? But try to understand the developing brain, right? We are trying to see how the pieces of the puzzle are connecting, and different parts of the brain are trying to get online. But one of the things that, again, I said is that we don't really understand the biological mechanisms that underlie, that make this, that explain how these early adverse experiences get under the skin to unfold during development and lead to these alterations, these anxiety, depression, etc. And this is because, again, for that, you really need to perform prospective longitudinal studies. And this is where, and I will read you the testimonials of these researchers, we really need translational animal models that have ethological construct predicted validity, and that are important to understand the developmental mechanisms that underlie the alterations in the brain and in social cognition, even how does attachment, right? How do you learn since very early on that you cannot trust anybody? And for this, really, we look into animals to do these studies that require prospective longitudinal studies with experimental control. Now, we have learned a lot from rodent models of early adverse experience, and I'm going to be defining during the talk today early adverse experience alterations between the interaction between the mother and the infant and maternal care. And what we have learned, and we have learned a lot, is that maternal separation, particularly intermittent maternal separation in rodents, has a whole set of array of alterations that mimic really and they are similar to what you see in humans. And you can see that in adults, you see increased anxiety, fear, and aggression. You can see anhedonia and the impaired reward processes that link these animals that experience intermittent maternal separation with increased risk for drug intake. You also see increased stress reactivity, activation of stress systems, and so on. So these are very, very similar to what you see in humans as long-term outcomes in related to psychopathology. Not only that, but we have also learned that from the rodent work that this is what is so-called the maternal mediation hypothesis. This happens because there are mainly behaviors of the mother that are not happening, that are necessary for the developing brain, and they have identified a particular set of maternal behaviors that are very important to regulate the proper development of networks that are involved in the regulation of stress and emotional reactivity and reward processes. And the main behaviors that have been identified in the rat, in particular, and also in the mice, in the mouse models, is that licking and grooming the pups and particularly anogenital stroking, are critical for the proper development of these animals. So these are the maternal behaviors that have come out from the rodent literature as being critical. Of course, archback nursing, because that is the feeding component. Now, of course, we're going to need to do an evaluation of how much can we actually pull from that literature, because there is a complementary value on the non-human primate models. And one of the things is that there are some things that rodent models don't model very well, and one of them is the complication of the relationship between the mother and the infant and the maternal behavior of primates. First of all, rodents, rats in particular, so the weaning and of the pups happen at some time, like 21 days, 25 days, and at some point they just break off from the mom. Now we have a relationship with our parents that is has a lifetime duration, right? We all are paying for college, or just, the kids depend on us a lot. Well, rhesus monkeys are the same. You are a female, you stay in the group with your family, and the relationships that you build with your other family members and your mother are critical for your survival, right? So not only that, but there are what we have to think is about the behaviors that have been discovered and mapped in the rodent literature as being critical for proper development of the brain of the rat pup are licking and grooming and genital stroking. I don't know about you, but I didn't do that to my child when I just changed the diapers. And that happens because for a rat pup to urinate or defecate, the rat mother has to actually stroke the anogenital region in order to eat, for defecate and micturate. Primates are born in a more developed state, so we are, thankfully, we don't have to do that really, really nasty task. So, but this is a very important point, right? Because we are talking about, we are having a lot of eye-to-eye contact, we're having a lot of proximity. Yes, we have physical contact that stimulates the release of serotonin, oxytocin, and so on. But it's much more than that. Just the presence of the mother we have published that can buffer the stress responses of the infant macaque. Just the presence, the physical presence. And there are results published, findings published by Seth Pollack's group, where even just a phone call from a mother can actually buffer the stress response of a child. So we're talking about much more complex set of interactions for much longer that cannot be completely modeled by the rodent literature, by the rodent studies. The other thing that happens is that there are some alterations that had not been modeled very well with the rat or the mouse. And this is, for example, alterations in basal stress physiology, like diurnal rhythm of cortisol, is not very well mapped. There are a lot of alterations that you can see in the human literature. There is a flattening in the diurnal rhythm of cortisol that people have described in children that have undergone childhood maltreatment or other adverse experiences. And that has not been very well mapped by the rodent studies. And this actually can be explained because the rodents are born earlier in their developmental, in the gestational state, right? So they are born blind, they cannot hear, the eyes have to open, the ears have to open. So what we think happens is that the HPA axis, the hypothalamic pituitary adrenal axis that control the release of stress hormones, is actually born at the later stage in primates than in rodents, and is not able to model some of the alterations that you can see in humans. And that's why we are turning to macaque models. And the other thing that has come out today is that the effects on prefrontal cortex development, of course, a species that is closer in not only the connectivity, the cytoarchitectonics of the human, in terms of the human prefrontal cortex, but the neurochemistry, and we have talked about acetylcholine, but I'm going to be talking about other neurochemicals that are differently distributed in the human, in the primate, and the rodent. When you're trying to map the effect of stress on the development of the prefrontal cortex systems that are showing up expressed in different regions, you're not going to have the same set of alterations. Here. Just to give you, just to pull from what Anita was saying earlier, which was really, really, it was deja vu for me because I had a similar type of experience with stress neuropeptides and mapping them in the primate brain. It doesn't look very much in some regions, like the mapping in rodents. And so you're trying to study the effects of stress on stress systems, and they are showing like stress neuropeptides like corticotropin releasing hormone is expressed in different places in the amygdala. If you're trying to study the development of that, definitely a rodent model is not going to be a good model. The other thing that we have been mapping is the expression of the glucocorticoid receptors in neocortex and hippocampus. And one thing that I also had a lot of problems when I was trying to publish this journal neuroscience paper is to try to demonstrate to everybody that the glucocorticoid receptor is really expressed in a different in different places and with a different level of expression in the macaque hippocampus and prefrontal cortex than in the rodent. And these type of studies have to be done. We really have to put a lot of effort in the translational from one species to another, because if we're going to be studying things in the grown place and with the grown system, with the grown neurochemical system, is really not going to get us very far away. So this is just one of the contributions that I wanted to provide here to this symposium of Colloquia on why we really need to use non-human primate models to address some of these questions that are developmental and related to stress physiology or stress experiences. Okay, now, why are we focusing on childhood maltreatment? Well, it is a devastating experience, everybody knows here, right? It is a major public health that is really probably affecting many more children than we think and that are reported, because again, many of that goes under the radar. And of course, you have the risk of being killed or really having brain trauma, but there are also emotional scars, right? Like what we hear over and over again, and is the effect of having these traumatic experiences and then having effects on your emotion, in your relationship with others, your attachment, and in your physiology, the way you can regulate stress and emotional reactivity. So we know that this is happening in humans big time, that childhood maltreatment is a major risk factor for the development of depression, anxiety, drug abuse, problems with attention. I already mentioned that. But we also know that the earlier it happens, the long-term effects are even worse. And this is actually very, very, very serious, because not only that, but there are a lot of children that are experiencing maltreatment early on. So this is a huge problem, because there are many, many things developing. These are critical periods for the development of the brain, for the development of our social cognition, attachment, etc. So as I said, the human researchers look at us to try to address, like, how is this happening? They know that there is the link, but they don't know what is mediating that link. What are the biological signals that are mediating the effect of an early adverse experience and turning it into alterations in brain development? So one of the things that is very important to point out is that childhood maltreatment is not just a human problem, because as I show here, has been also reported in non-human primates, and this is both in captivity and in the wild. And this happens with the prevalence rates that are similar to those in humans, 2 to 5% of the population. So we really have here a unique opportunity to use this naturalistic model of infant maltreatment to do prospective longitudinal studies that can be done since birth and with a strong experimental control, so that we can actually address this question of what are the mechanisms. And one of the things that we can do that researchers that study humans cannot is that we can do randomized assignment to group. We can cross-foster rhesus monkeys. It's very difficult, much more difficult than doing it in rodents, but can be done. We have done it. Then we can follow them longitudinally, and we can study them from one generation to another to see things like transgenerational transmission through, for example, epigenetic mechanisms. So huge opportunity to study these animals. Okay, so this is our subject, the macaque rhesus monkey, also known as macaca mulata. And one of the things I wanted to point out is that one of the reasons, these are the reasons why we are studying these animals. We selected this species because it's highly social and it forms very strong mother-infant bonds. And not only that, but these bonds are lifelong bonds. If you are a female, you have your mother forever until she dies. Maybe you take over being the boss in the family because they live in families where they take over, like who is the matriarch. The other reason why we study these primate species is because of the huge amount of information that we have on the developmental impact of early social environment. And the social and biological similarities, the well-developed brain, like including the prefrontal cortex, resilience to disease, and so on. But one of the things I want to remind you all is that this is not trivial, because when we talk about all the information that is already existent on the huge impact of social environment on the development of primates, we forget that much of this was developed by Harlow, and that Harlow's studies with the deprivation, like pulling the infants from the mothers, even if many people right now may be completely considered very cruel, and we forget that that changed the way we raise our children. We forget that in the 50s, the pediatricians were recommending mothers to put the babies down in the crib and do not pick your kid because you're going to be spoiling it. That there was this distance between the parents and the infants, and there was not a lot of contact, right? So Harlow's experiments really changed the way we developed and we treat our children. Now you see people with snugglies, carrying the babies everywhere. There is a lot of contact, eye-to-eye contact. So it definitely had a huge impact in our society. The other thing that we also forget is that macaques were used to develop attachment theory. That was Bowlby, John Bowlby. And that was huge too, because that's what told psychologists, like particularly children, developmental psychologists, that the formation of an attachment with a figure early on is critical for the proper development as an individual in society. Okay, I don't know how I'm doing on time. Oh, very little. Okay, so I don't care if I don't go through the main, through findings or whatever. I just want to hit the main points so that we can at least cover what is the state of the field on this. I wanted to cover like a little bit of the developmental milestones on this species, although some of you know, because this is another point of translation that is really very important, that these animals go through developmental milestones that are similar to those in humans. One of the things that happens is that early on, you have this mom and infant that are in contact. They are in very, very physical contact. And that happens until weaning, when the infant is going to be exploring, getting off mom, and just coming back to her when he gets scared. And it's not until then that social fear emerges. And this is actually very similar to what you see in humans with stranger anxiety. Right? So we have not only the brain development is similar, not only mother-infant interaction is very similar in the two species, but some of the milestones that these animals go through are also very, very similar, which gives us also times in the development that we can actually study processes that are important for humans to understand. So we study these animals who have these social groups that are present in the breeding colony in Yerkes, where we can actually study animals in their social environment. And these are complex social environments where you have these big family groups that live in these social strata, from high-ranking animals to low-ranking animals. This is maintained by aggression, so there are like high-ranking, alpha, and the ones on the bottom that also are experiencing a lot of stress. So you can actually mix in these studies not only the effect of maltreatment, but the effect of being in a low socio-economical status or undergoing a lot of aggression by others, or bullying, if you want. Okay, so this is a summary of what this infant maltreatment model is, in the way we have been developing, well, it's a naturalistic model. We are not doing anything. We're just characterizing it, and we have established the validity, contract, predictive validity, etc., face validity. So again, involves physical abuse, which is this violent behaviors exhibited by the mother towards the infant. And then it's also comorbid with high maternal rejection very early on in the first 3 months of life. And these are aberrant behaviors. They are repeated from one infant to another. So this is part of the maternal trait. And it happens very early on. It happens during the first 3 months of life, and then they just stop because the infants get weaned and they just get on with their lives. There is intergenerational transmission of these phenotypes, which makes for a very interesting study of epigenetics, right? Okay, so these are the rates of abuse and rejection that the animals get. So this is just one of the several studies where we have replicated these findings. As you can see that the first month of life, the animals that are maltreated, they get a lot of abuse, 1.5 rates per hour. And then it drops by the third month of life, and this is basically because the infant can run away from the mother. It's strong enough that at that point it's saying, I'm not going to be dragged through the compound. But then there is this other comorbid aspect of the maltreatment, which is the rejection, where you can see also very high rates, and it's not exhibited by the control mothers, and you can see how high they are, right? 1.5 rates per hour, and are happening at very high rates, and they are staying on for a long time. So one of the things that, just going to give you the bottom line here, is that what we have seen is that we are studying the contribution of these two different aspects of experience, and we have found that this is actually more detrimental for long-term outcomes. We see that this is receiving rejection early on is actually a worse predictor for developmental outcomes. And this has been replicated through different studies. And one of the things that we also know is that this is indeed stressful. It's a stressful experience because we have seen increased accumulation of cortisol in the hair of the babies from birth through the first 6 months of life. So during these first months of life, you see high accumulation of cortisol in hair. Other things that we are seeing is, of course, a lot of increased emotional reactivity. We see tantrums, screams. These animals are really, they show high levels of anxiety. But one of the questions that we had is like, okay, this happens during infancy. Is this long-term? Is this staying through adolescence? Is this really mapping into what people see in maltreated children? And the answer is yes. Actually, we see that these are persistent alterations in emotional reactivity and also increased social aggression. But some of these effects actually get magnified with age and emerge during adolescence, which is something that is very common knowledge in the literature. And what we are seeing also is this comorbidity between internalizing and externalizing behaviors. So just to come back, because there is not a lot of time to go through the specific data, but just to hit the two main points that I wanted to make today is that we have been identifying biological mechanisms that underlie changes in brain development. And one of them is that we have seen alterations in the development of the brain serotonin system, which is, of course, involved in emotional regulation, impulsive aggression, etc. And we are seeing that the animals that are maltreated have lower levels of serotonin, not only in CSF, which is shown here in serotonin and the metabolite 5-HIAA, but there are myriad of changes in the 5-HT receptors, particularly in the prefrontal cortex and in amygdala. So this is interesting because it's also predicting the levels of anxiety that these animals have. So this actually maps very well as a translational result in the human literature. But what I wanted to tell you today is about some of the biological mechanisms that we have identified. And that actually what we have seen is that the alterations in brain serotonin are not really predicted by changes in cortisol, but by increased inflammation that is happening early on during development. Right now we know that inflammation is another stress-induced alteration, And it's another pathway by which you can alter a brain, and that is linked to anxiety, depression, etc. But when we published this, was really novel information. And then just to finish, the other thing that we have been finding is alterations in prefrontal amygdala and prefrontal striatal connectivity. What we are seeing is that the animals have, throughout their lives, a weakening on the prefrontal amygdala and prefrontal accumbens connectivity, which is very interesting for drug abuse. Okay, so we know, as Jocelyn said, that this, yeah, I'm finishing. The vulnerability of this prefrontal amygdala connectivity, these networks, and the connectivity with the nucleus accumbens and serotonin is due to the fact that these systems are developing very fast during early development when the maltreatment is happening, which is during the 2 to 3 first months of life. So that's something that we know, and I just wanted to finish, since I have to finish, I want to read you the testimonials from these 2 researchers, but I just wanted to leave you with this take-home message, which is basically telling you something that you may know. Maternal care is critical for proper brain development, but with the use of non-human primates as a translational model, we are really tapping into what is specifically important for the development in the primate. And this is very important so that we can actually tap into the development of treatments for anxiety, depression, and drug addiction. And here it is, like some of the testimonials from Megan Gunner, which is the director of the child development in University of Minnesota, who is bringing up the memory of how important Harlow experiments were, an attachment theory that was developed in monkeys. And what basically both of them have a similar point, which is that they cannot do these studies in children, and that they are actually, without this animal work, particularly non-human primates, they are flying blind because they don't know where to start. They cannot study these kids. Another thing that Mary was saying is that because of the homology with the monkeys, including the focus on attachment, the power of randomized assignment to conditions, which we can do in monkeys, and they can't. It's so important to understand the consequences of these of specific conditions too, like what is the specific effect of poverty versus neglect, abuse versus neglect, and so on. And this is just to thank everybody that was involved in the studies. Done. Okay.